Good afternoon, everybody. I may have seen many of you in our stand, but I've seen so many faces and heard so many names, I'm afraid I can't remember all of you. My name is John Mulder. I'm the CEO of Diagnoptics, and I'm going to talk about advanced glycation end products, or AGES for short. If I have to mention the long version, we'll be running out of time in a minute. The presentation I put in had the title, How Ages Kill Longevity. And we'll see in a minute how that is the case. By the way, this is the device that we have developed. So what are ages? Ages are dangerous compounds that form when sugars react with proteins. Now that is a process that just happens in your body. It also happens when you're cooking. But in a number of steps, steps, they form these end products. I won't bother you with the Schiff process and the Amadori products. There's plenty of research available to get into the details, since we only have limited time. Once these ages are formed as end products, they will cross-link to and accumulate in your tissue. And that can be various types of tissues, protein-rich tissue. Like, for example, your heart tissue, blood vessels, kidneys, your brain tissue, your cartilage, and also the skin. I've highlighted the skin, and that you will find out why that is. We could say that ages are the linking pin between many chronic diseases, because this cross-linking and accumulation to tissue will damage that tissue. It will stiffen the tissue and the tissue will lose its function. And therefore it is associated with diabetes and its complications, with cardiovascular diseases, with renal failure, kidney disease, you name it. And recent studies have even proved it is associated with Alzheimer's disease and some sorts of cancer. Now, I always say that ages have only one good characteristic, and that is that they are fluorescent. And that is the principle that we use to measure ages, because ages tend to fluoresce when we shine a tiny bit of light on them. Now, shining light into skin tissue, there it is, skin tissue. Skin tissue is an extremely complex type of tissue. So when we shine a tiny bit of light on the skin, in the skin, first of all, what happens is that the surface will reflect part of that light. Also the hairs, you see a bit of hair. Hairs will also reflect the light to a certain extent. Oops. Right, let's leave it there. Then part of the light is absorbed by the melanin in your skin and the hemoglobin in the tissue. Some of it is scattered by molecules in the tissue, and the remainder will excite the ages that are cross-linked to your collagen, and they will leave the skin, and that is what we capture with this device. And this, here you see an inside of the age reader, as we call it. So we shine a tiny bit of light in your skin, you put your arm on the device, and these ages start to fluoresce, and that is picked up by the sensors in the device, right? Now, obviously, we spend quite a lot of time and money on research and validation of this particular methodology. In the past, we took biopsies from the dominant arm around this spot of the arm. Why? When you come to me and do a measurement, I always ask you for your dominant arm, because that is where, collagen is not evenly distributed, and it can vary from dominant arm to non-dominant arm. We want to stay as closely as possible to the validation point. And here are the results for that validation study. So you see the pentosodine, which was analyzed in a laboratory, compared to the autofluorescent signal of the same persons. That has a very good correlation. But then we only know that we can measure using light and fluorescence. We then did a study to see how does the age level vary with calendar age. That is what you see here. And again, a very strong correlation and a pretty straight line. We did validate it from 0 up until 80 years of age. I suspect that when you grow to 100, your level would be quite low, because otherwise you would have been dead already. So here you see the graph that the device shows you. And this is an example of a person 44 years of age with an AF, stands for autofluorescent, level of 2.9. Now the scale, the vertical scale, is what we call an arbitrary unit. So it is not something like grams or milliliters or millimoles or anything like that. Let's say we just invented that scale. Now you see 3 lines. The middle line is the average value, the sort of reference line for healthy individuals. And the top and the bottom represent the standard deviation. So as long as you are within the top-bottom area, you're pretty much okay, right? Now this particular person has a 2.9, and that is touching the red zone. And from a lot of research, we know that if you're in the red zone, you have a significantly higher health risk. If I know nothing else of you, I cannot say it will become cardiovascular, it will be diabetes. We need to do more research, but I can say your risk is higher than average. Okay? So I hope we have some time to go through a case study. This is an actual case study. A diabetes patient, a 44-year male, a pilot, 1 meter 74, a bit overweighted, you might say, with 94 kilos and 174, and a BMI of 31. And he's been a type 2 diabetes patient for already six years. Obviously, the man is consulting his doctor on a regular basis. He gets these medications. Here you see his HbA1c scores, his cholesterol scores, and this man has no symptoms or does not feel any complications. He thinks, okay, I have diabetes, but that's it, right? So the clinical conclusion with the current guidelines is that this patient is a well-controlled type 2 diabetic who is not at risk of developing any complications. So your doctor might say, well done, mister. Keep on doing what you've done and see you in 2 months. Okay? However, he had a very clever doctor, a sensible doctor. He performed an age reader measurement. Now what happened? This was the result of this patient's measurement. A 2.9, as we've seen before, indicating a seriously higher health risk. That was the signal for this doctor to perform additional tests. So the score in this case that here is a definite cardiovascular risk happening. First of all, he did a cardio stress test that came out negative, but the IMT measurement came out positive with a 70% stenosis. So a clear case of arteriosclerosis, and that has never been found before. And why was that? There was no reason. The current guidelines did not look for it. So there were no indications of any risk whatsoever. So this patient got some surgery, and now he was fully recovered and back to work as a pilot. Should his doctor have not done this age measurement, it could have been fatal. So the age reader measurement was in fact essential in detecting this life-threatening cardiovascular complication from the diabetes. And we know that diabetes patients that are not in control have a much higher risk of developing complications, and these are scary, as you may well know. So the clinical use of the reader, and I'm almost done with my slides. you can have a good indication of the complications in patients with known conditions that we've just seen. Very important, use in preventative care. It's a very easy, non-invasive way of assessing health risks. A single measurement only takes 12 seconds. And we are also doing some research this spring. We will do research in the United States on the perioperative risk assessment. There are already some researchers available on the internet that have proven that ages perform as a very good biomarker to assess the perioperative risk. And that basically concludes my presentation. Any questions?
Hi, so I'm an ENT surgeon. So it caught my eye that perioperative risk assessment. So how are you using AGES in that?
We are using AGES. AGES is also a sort of a biomarker for frailty. You can use it in elderly patients, but it will also be used in perioperative risk. I mean, if you before you cut a knife in a person, you want to know if that person is capable of surviving the surgery. Okay. And I know of one study that has been done by a PhD student in Holland, researching the use of AGES as a biomarker in cardiovascular surgery. And that came out that AGES had a very good predictive value in the risk of the surgery itself, and also in the 30 days after the surgery.
Post-op recovery? Are you like predicting that? Post-op recovery?
Yes, also, yeah. So the risk during the operation, but also any risk of complications in the 30 days after the operation. Yeah?
Can I access the study? Something like it's already published? Or?
Yes, that is already published. Yeah, yeah. It's Rosa Smoor. Difficult in Dutch names, but okay. Come to me and I'll guide you to it. Yeah?
That's a good question. How many people are using it? How many hospitals are using it? Anyone in India?
In India, we have just started to bring that to the market. Across the world, over the time that we are active, I think we have sold more than 6,000 units across the globe. I think that Japan is probably our biggest market. And I think that's also due to the fact that our Japanese colleagues are very active in educating the clinical world. Because to be honest, I think that too many clinicians know about AGES, what they do and what you can do with the measurements. So education is a first step.
FDA approved?
Not yet. We are working, as I said, in the US, we will start this spring with the clinical research. We're pretty confident that 2 years from now we have the FDA approval. And also another case study that it is a good biomarker in the perioperative area.
I was just curious, how do you account for the non-specific fluorescence? Probably you mentioned it and I have missed it, but there are other molecules in the blood that
Yes,
would also...
Yes. We know that, I didn't include that slide because I had only 15 minutes, but we know that AGES excites fluorescent signal in a very specific bandwidth. So that is the bandwidth that we measure and nothing else.
So you don't get any interference from...
No, no.
That's, okay.
Also, it has to do with the bandwidth of the light that we send in the skin. So that is a very controlled bandwidth, a narrow bandwidth. And then again, the excitation bandwidth is also quite limited.
And the cost?
The cost of the device we have several business models here for India. So you can buy it outright for a fixed price. You can also lease it for 1 year, 2 years, maybe 3 years. Or you could adopt a pay-per-use model. That is something you have to discuss with Hospimax. Hospimax is our distribution partner here for India. They're based in Delhi. But the outright price for outright buying is 15 lakhs, 15.
Okay.
How reflective of the skin measurement with the overall, all the organs, like you are measuring using the skin, but we can cross the barriers, the brain and other organs. How reflective the skin measurement with other organs?
Yeah, I had a couple of questions over the last few days that people say, okay, so you're only measuring skin quality. Basically, yes, we measure the level of ages in the skin, but we know from lots of research that the amount of ages we measure in this spot is representative for your entire body, for your blood vessels, for your heart, for your kidneys, and also for your brain tissue. So through lots of research, we cannot say this measurement is for your collagen, this measurement is for your brain tissue. That is not how it works, but through lots of research we know what the association is, what the correlation is, what the risk is with age levels and certain, and even for Alzheimer's disease, for example.
So, what is the reference instrument that you use for age?
So the reference,
Reference instruments, like where you compare
all right,
the actual
well,
results.
as I showed you on the slides, maybe I can put it up.
Maybe I can check with you later. That's also should be fine. And what is the competitor do you have in the market?
We don't have any competitors.
Okay, then
We
then
are,
for applying
at least
for
we are,
FDA,
we are the only one using this methodology.
sure.
Coming back to your first question, we did some biopsies and analyzed the biopsies in a laboratory and compared that to the fluorescent signal. And that is like your reference device, right?
Okay, so for applying 510K for FDA, you have to use those reference measurements, right?
Yeah, FDA is a whole different matter. We'll talk about later. Sorry, again?
With the next version, have a better display.
Yes, absolutely. Yeah, a more modern display. Yeah, absolutely. Thank you. Yeah.
Thank you so much. Very nice innovation. I think we'll cut the questions.
Yes, very good. Yeah,
We can.
we are validated Fitzpatrick 1 up until 4. We are working to measure darker colors as well, but we need.
So please, can we? No, we can have the discussion later, please, during the coffee session. Let's stop the questions. Thank you so much.
I've tested.
Sure, but in the interest of time, we have to move on. So, no. It's. Let's have the discussion much later.
Yeah.
Very interesting, very nice innovation. Thank you so much for the presentation. Let's put our hands together. Thank you.